Can you hear me?
Yes.
Okay. Good morning, everyone. Thank you very much for inviting me to present some research work and control program for parasitic diseases. Actually, today I would like to talk about the hand-mint infections, not on parasite diseases in Laos, but only hand-mint infections. So the first slide, I would like to, this slide show you the priority hand-mint infections in Laos. It's composed of 4 parasites. The first one is lymphatic filariasis, and the second one is foodborne helminthiasis. This we will especially for Opisthochis viverrini. And the third one is blood fluke. It's Sistersummamekong. And the fourth one is STH, soil transmitted helminthiasis. So let's talk about the lymphatic filariasis. Sorry, I want to move this. So this lymphatic filariasis, we start to survey in 2002 because and after that 2007 lymphatic filariasis were found only in Phu district in Attapeu provinces, from 2% to 28% infection rate. And after that, in 2009, lymphatic filariasis cases were confirmed in just the district of Attapeu provinces, not only Phu district. So we extend the survey for filariasis in Sanam Sai. That's we found 27.4%, and Phu 17.6%, and Sai Setha district 11.1%, and Sama Kisai district 4.4%, and Sansai district we found 1.9%. You can see the map of lymphatic filariasis in Attapeu province. You can see this one, the green one, that's it means that not endemic, and the red one is endemic area, and the yellow one is the new endemic area, and you can see the percentage in each district. So in Laos, we confirmed that the lymphatic filariasis belong to the Wuchereria bancrofti species. So, and after that, in 2013, the neighboring provinces were surveyed. We found in three provinces, Sekong, Champasak, and Sarawak, but we found only in Sekong province with the percentage at 0.33%. This slide shows you the control of lymphatic filariasis. So the main activity for controlling this parasite is we conduct MDA with DEC plus albendazole. This is only at the province with age from 2 until 65 years old, that we provide the MDA on this age group. And after that, we expand more MDA in 2020 to 2017 in 5 districts in Attapeu province, this Sanam Sai, Puvong, Setha, Sai Setha, Samaki Sai, and San Sai district. The MDA coverage is around more than 65% among the total population of Atapu province. Also, we do the surveillance for blood examinations and also for the vector control. This control will integrated with malaria control program of the country. So next slide for Sistersummamekong. This is show you the history of Sistersummamekong in Laos, which we filed the first case report in 1957, and after that, 1918, Lao government realized that Sistersummamekong is the public health problem in Laos. So we conduct many survey, and also we perform the MDA implement with health education with support by WHO and the government health pharma fund. And at the period of time, the infection rate reduced from 50% to under 2%. And the map shows you the endemic area of Sistersummamekong in Laos, which occur only in the south of Laos, only in Champasak province. You can see this, Champasak province, and only 2 districts in Champasak province. There are Khon Island, Khon district, and Mulapamuk district, only this one. And this is the endemic area of Sistersummamekong in Cambodia. Next slide, just brief you the history again. The MDA was stopped in 2000 to 2003, and after that we resurvey again among 64 villages of Khon district and 10 villages of Mulapamuk district, and we found that Khon district have the infection rate of Sistersummamekong is around 11%, and the high infection rate is 47%. And Mulapamuk district is the infection rate around 1%. And this is the intensity of the infection. We found that the moderate infection intensity rate around 30%, and high infection intensity rate is 15%. So recently in the data, the information from 2015, in some spot area, endemic area, was conducted the survey in Khon district. We found that the infection rate about 22.2%. This is we survey the case of we found the case of severe cases of Sistersummamekong in 2007 to 2010 with the severe in the adult persons, and also with children. We also found the severe case of Sistersummamiosis. So after that, in 2015, we conduct the transmission survey, and we investigate the host of Sistersummamekong. This is the snail intermediate host that we collect from Mekong River, and after that we examine them to detect the Cercaria. Also, we examine the animal reservoir hosts such as dog, cat, and pig, also buffalo. This is the result of animal reservoir hosts of two islands of Khon district. We found that dog infected with Sistersummamekong it's around 14.7%, and for cat, pig, and buffalo, we could not find any egg from their feces. About the snail, Neotricola aperta snail, we collect more than 29,000, and we found that 4 of them are positive with Schistosoma cercaria. It means that it's around 0.01% infection rate. Let's talk about the control notice for Sistersummamekong. From our observation and research, we know that low practice of stool examination in the endemic area, especially in Khon district and Mulapamuk district. Therefore, if we increase the behavior of stool examination of physician and lab technician in district hospital, it will be good for detect and treat the person who got infected with Schistosomum And as I show you some reservoir hosts infected with Schistosomum especially for dog. And the previous study also report for pig infected pig also got infected with Schistosomum. Even in my study, we could not find any pig infected, but we have the previous report also from pig. So it means that the reservoir host transmission could be a problem for elimination of Schistosomum. It will be a good option if we consider and do the One Health approach for the control approach. I mean, we have to do the control program among humans and also the animal, not only human population. So right now, we use in Lao, we use cattle-cut method as the technique to detect the egg of Schistosoma in the patient. And also we have some research for using the circulating antigen in urine and serum. This is we need to validate in the country pertaining on sensitivity and also cost effective. This is for the surveillance purpose to achieve the elimination of Schistosoma in Laos. Okay, let's go to Opisthocus viverrini. This the national scale survey in 2002 for Opisthocus viverrini. You can see that the high infection rate, more than 20%. It's the orange color. You can see from the central to the south of Laos. For the medium endemic area, the yellow one, and the low endemic area is the green one. And in this research conduct in the primary school children, not the whole, not the community population, and we can summarize that the population at risk are more than 70%, and the infection rate is from 0.1 to 7%, and we got 6 provinces. The infection rate more than 20%, and 7 provinces in Laos infection rate around 5 and less than 20%, and only 4 provinces the infection rate less than 5%. And this is the research group from Swiss and Laos. We conduct the map of predict prevention for OV infection only in Champasak province in 51 villages. But this is the community, the community study, not only primary school children. We can find that this is general in many color belong to the high infection rate. You can see like this. This is if the white one is less than 10% of the prevalence, but we almost are the red one in the Champasak province. We also collect snail and some fish to survey the intermediate host for Opisthocus viverrini. We collect many fish and Bithynia snail from the pond and some from the ponds and some stream of the Khon district. And this is the result of animal reservoir host for Opisthocus. So we found that dog infected Opisthocus viverrini is around 25%. In cat, it's more than it's around 53.1%, and in pig only 0.9%. We could not find any egg, Opisthocus egg in buffalo. The infection rate in snail, Bithynia snail, is around 0.3%. This is the fish that we collect and examine. Is around 600 fishes we examine, and 21 species. Among 21 species, only we found in 11. 11 species were OV positive. Is overall prevalence of 26.9% for the second intermediate host for Opisthocus viverrini. You can see the most heavy infection fish got infected in Hampala and Dispa. This is Pasud in Lao language that we collect 101 Pasud, and we found that around 87.1% infected. So from 2010 to 2007, in 6 provinces that got the infection rate more than 20%, we do Parasitic control MDA and with health education. And after that, after 2007 until now, there is no more regularly MDA in these province because of the budget concern. And also, if you would like to eliminate the Opisthogyridae, not only humans, because we also found the infections occur among the reservoir hosts such as dog, cat. So we need more very specific control approach to control this parasite. Okay, let's go to soil transmitted helminthiasis. In this map, show you the infection rate of STH infection is around 27.5 to 96%. So we divide in two color. The moderate endemic is in the center and south of Laos, and very high endemic in the north of Laos. You can see here, and in the south of Laos, only Sekong have a high infection rate for STH. For other parasites, for other helminth infection in Laos, it's like Nattostomas spinicerum and Strongyloides and Paraconimus. This slide show you the study for Nattostoma in Laos. This parasite is very few research on this, because there's not so much research on this parasite. This is I did in 2009, do a zero epidemiology survey for nattostomiasis with 3 provinces in Laos, Vonghachack provinces, and we found that 3.6% got infection rate for nattostomiasis. And from Vientiane capital, we got 47.1 in Vientiane capital, and also Champasak we have 38.6% zero prevalence rate in Champasak province. This is on, and we know that in Vientiane and Champasak got a high zero prevalence for Nattostoma in Laos. For Strongyloides, as you know, if we use only Catocat, maybe very low sensitivity for detect the Strongyloides. That's why we did the specific method for detect the Strongyloides using a Bergman test, and we found that in Khon Island, in one island, a long island, we found that 40%, 41% rate for Strongyloides, which meant was the high risk of infection. And we also found the children under 6 years was very high infection rate, and we suppose that humans and dog maybe share their Strongyloides parasite together, because of their children behavior in rural area of this island. They play with soil a lot when they play on the ground. So we need further genetic study on humans and dog Strongyloides. The last one is for Paraconimus species. the Paragonimus species is prevalent in the north part of Laos and also in the mountainous area. So in 2006, we did zero prevalence in Kasi district. Kasi belongs to Vientiane province, which far from Vientiane capital, around more than 100 kilometers from Vientiane capital. And before that, the zero prevalence of the population in Kasi district around 6.7. And we found the two active cases from sputum in this study. And we examine the sputum that the egg positive is Paragonimus heterotermus egg positive. We could not find Westermanni egg in human sputum in this study. So the paragonimiasis still could be sometimes misdiagnosis to tuberculosis in the district level. This is we have to concern about this for a TB program should in the mountainous area and in the north part of area should consider and keep in mind that can be misdiagnosis for paragonimosis and tuberculosis. Thank you very much for your attention.
Thank you very much, Dr. Youthanavanh. So we are now in the Q&A session, and we have one question in the chat box. One help would this approach would be nice to control the zoonotic disease, for example, schistosomiasis. How is your opinion?
Oh yes. Yes, because of we found that the transmissions dynamic, including the animal reservoir host, and these are hosts is domestic hosts that live with the people in the community. So that's why one help that I think it's very good for control approach for schistosomiasis. But one thing that I think we should survey for not why animal also around the area, endemic area also it will be useful.
Okay, thank you. So if we don't have more questions, so thank you very much, Dr. Youthanavanh.
Thank you.